Good morning students. Today I am going to discuss a very important topic on apoptosis. Apoptosis is programmed cell death. It is also called as cell suicide. So, programmed cell death. So, our body cell is planning to get killed off. So, it is pakka programmed, that is called as apoptosis cell suicide. Then there is another term called necrosis. So, now itself I will compare apoptosis and necrosis. Necrosis is cell murder, which is abnormal, because here what happens is, I will just compare apoptosis and necrosis. Apoptosis is programmed cell death. Cell wants to die because of some reasons which I will be explaining, but here unexpected cell death, which is cell murder, and here there is no inflammation. Please note it down, which is very important. Here why necrosis is bad? Because of this necrosis inflammation occurs, and here no membrane rupture, or membrane is intact. Cell membrane is intact even if cell is committing suicide, intact. Here rupture of cell membrane takes place, and because of the rupture of cell membrane only some particles or debris comes outside and it infects other cells also. Yeah, it spreads. Here no spread. And another important point in apoptosis, this is shrinkage. How the cell gets killed, the cells will shrink. That also will explain how the cell is being killed by the process of apoptosis. But here the cells get swollen. So, necrosis is bad, apoptosis is good. Now, next question, very important question, why apoptosis has to occur? Embryological, very important. For example, embryological development cannot be complete without apoptosis. For simple example, in the embryo, these spaces will not have this. For example, see thumb, index finger, middle finger, all those things, and I am having clear this space is more, the gap between this thumb and this is more, this is less, and I am having free space, or there is no web between this. You see in duck, there will be that webs. We humans do not have these webs, but in embryo we might have had this. Then how this gone? Why you are getting the space? Yeah, you know the answer. The reason for the space between these two fingers, the space between these two fingers is apoptosis. So, it has been programmed that I should have space between each finger, and that space is created by apoptosis. So, the embryological is one reason. Second one is thymus involution. Many examples are there, I am just giving an idea. So, in the embryo thymus is well developed, but as the adult matures, thymus size decreases. How thymus size decreases? Apoptosis. Okay. Now, all this is physiological or pathological. Otherwise, apoptosis, as far as we know, it is for good to the cell. As far as we know, apoptosis is good for the body, but that can be divided into physiological apoptosis, pathological apoptosis. Pathological apoptosis means, if our body is attacked by some bacteria or some virus, what do I want to do? That cell which is attacked by bacteria, I have to, or our body, our cell, that cell has to undergo apoptosis, so that the cell will not spread to other cells, so that the cell is destroyed and our body is protected from infection. Then that is pathological apoptosis. So, these two comes under physiological apoptosis. Now, I am going for pathological apoptosis. Example, any viral infection. Example, hepatitis. Any infected cell can undergo apoptosis. Then pathological, DNA damage. Cell can undergo apoptosis, so that the damaged DNA will not be replicated and be spreading. Then any misfolded proteins inside the cell, there also apoptosis is required, so that all those misfolded proteins is of no use. So, I have to destroy that. Next, how apoptosis occurs? It is with enzyme cascade. Main enzyme is caspases. As the name suggests, it is going to produce nucleases which will break down the nucleic acid, proteases which will break down the protein. So, the genetic material which is present inside the cell are broken down by these nucleases, proteases. For these enzymes to be created, caspases is needed. Caspases is nothing but C for cysteine, as for aspartate. If we take a protein, when this caspase comes to that protein, that amino acid chain is disturbed, because this is going to break down the cysteine, this is going to break down the aspartate. Then what will happen? That protein is going to be not functioning anymore. So, this is how the caspase is going to produce nucleases and proteases which will break down the nucleic acid and break down the proteins and thereby destroy the cell. So, under that caspases we have two types, initiator, note it down, initiator caspases, executor caspases. So, initiator caspases will not produce this, or initiator caspases is not going to do the breakdown, but it is going to initiate the process. Caspases 8, 9, 10, they come under the initiator caspases. Executor caspases 3, 5, 7, 3, 5, 7, and this is the one which is going to do or produce nucleases and proteases. But for this 3, 5, 7 to form, I need initiator caspases. So, there are some factors regulating apoptosis. Always remember, all cells cannot undergo apoptosis. Again, then someone can ask me, sir, then every cell will have this caspase, every cell will have this caspase 8, 9, 10, and that will help in 3, 5, 7. How, sir, why all cells does not undergo apoptosis? Yes, there is always a balance between life and death of a cell. So, yeah, this is a cell, this is life, this is death of a cell. So, when death of the cell will occur, already I discussed when apoptosis will occur. If I want to go for apoptosis, some factors are there which we call as life. I will put it here, anti-apoptotic factors which helps in life of the cell. Apoptotic factors which helps in apoptosis, which is death of the cell. BCL2, BCLX. Cell apoptotic factor is BAX, BAK, BAK. Actually many are there, I do not want to confuse, so I am just restricting with two. Anti-apoptotic factors which helps in the life of the cell is BCL2, BCLXL. Apoptotic factors which is helping in apoptosis so that that cell is died is BA. Remember A for apoptosis. So, BAX, BAK. There is always a balance between pro-apoptotic factors and anti-apoptotic. I can even mention this as pro-apoptotic factors, anti-apoptotic factors. If the cell is normal, then this will be more. BCL2 and BCLXL will be overtaking. So, there is always a balance between these two factors. Now, let us go for mechanism of apoptosis, which most of the people get confused. Everyone knows apoptosis means programmed cell death, cell shrink, no inflammation. But when you go in depth, many get confused. Even if it is given in first year textbooks, most of them do not understand the concept. So, today's video, now only we are entering into the difficult topic, mechanism of apoptosis. Already I gave an idea, caspase activation, but it is again divided into two pathways. Please note it down. Intrinsic pathway for caspase activation, extrinsic pathway for caspase activation. Intrinsic means the stimulus is within the cell, within the cell. Extrinsic means the stimulus for apoptosis or the stimulus for caspase activation is outside the cell. Now, I will go for intrinsic. So, this is the cell, this is the mitochondria. Now, just to concentrate. So, this is normal cell. We all know within the cell we have ribosome, nucleus, mitochondria, all those things. Now, we have drawn the structure of mitochondria. This is outer membrane, this is inner membrane, and within this two membranes there is cytochrome C. Just concentrate, the cytochrome C is normally present between the two membranes of mitochondria. It is not going to come into the cytoplasm. Normal cell, in a normal cell, the cytochrome C is present within the mitochondria or in between the outer membrane and inner membrane of mitochondria. It will not come into this is cytoplasm. If there is a decreased growth factor or the cell is not able to grow or if there is damage to DNA, all these are examples for apoptosis to take place. There is going to be imbalance. Now, here what happens if there is any stress? We call this as stress response, either decrease in growth factors or DNA damage or anything which is affecting the cell. Stress. Imagine the cell is under stress. What I should do? Whether the cell should be there or it could be destroyed. So, whenever the cell is under stress, any stress response, what happens? There are stress sensors. So, that is very bad to the cell because of this DNA damage or decreased growth factor. We call that as bad beam. Maybe the cell is becoming, for understanding I am telling, cell is becoming big instead of normal size, beam boy, beam boy. So, BIM, beam, Noxa, NOXA, Puma, the shoe name. So, all these are the sensors which gives the signals that the cell is under damage, stress, they are called as stress sensors. So what they will say, better we can destroy this cell by apoptosis and prevent from this cell to be committed to destroy other cell, preventing the infection to spread to other cell. That is one of the main aim of apoptosis. So now just to concentrate, this is very important. So now what happens here, you can tell him whenever this is getting stimulated, bad, beam, noxa, puma, all these are stress sensors. Whenever this is there, what I told first, there is always a balance between anti-apoptotic factors and apoptotic factors. They both are like same. But here what will happen? Which is going to be stimulated? Yes, back, A for apoptosis. So apoptosis is going to be stimulated BCL2, BCLXL. These are anti-apoptotic genes. This is going to be inhibited. So minus anti-apoptotic. So this is going down and this is going up. So apoptosis is going to take place. And all this, what is happening within the cell? So this is intrinsic. So what happens? Because of this, there is a channel created. Please note it down. Very important. Channel or gate. So there is a channel or gate created for cytochrome C to come out. So for this only everything is occurring. See, cytochrome C is actually present within the two membranes of mitochondria, but here a channel or gate is created. Now next, why channel or gate is created? Because pro-apoptotic factors are stimulated. Why pro-apoptotic factors are stimulated? Because stress sensors are stimulated. Why stress sensors are stimulated? Because the cell is under stress. Why the cell is under stress? Because decreased growth factor or DNA damage or even embryological nature. For example, if I want to remove this web space, there is a need to adjust the cell, because in embryo the space will not be here. So their cells are going to be here. I want the cells to be removed by apoptosis. So here all these will occur and this cycle is going to occur. Now just to concentrate, we are ready to complete. So what happens? The cytochrome C comes, I will write it here, extrinsic, we can go for the next slide. Cytochrome C into cytosol, note it down. And this one bind with APAF. If anyone wants to answer what is the full form of APAF, you can pause the video and answer. APAF. So APAF is nothing but apoptotic. So that means it is going to help in apoptosis. Apoptotic. P for protease. We already discussed. What is protease? Protease is an enzyme which is going to digest the proteins. Obviously, you are going to kill the cell by suicide. So apoptotic protease activating factor. Just here, this is the intrinsic pathway. Already we explained when intrinsic pathway for apoptosis takes place, the reason. So here the cytochrome C comes out. As the cytochrome C comes out into the cytoplasm, it combines with apoptotic protein activating factor, APAF. When this complex occurs, that will convert pro-caspase 9 to caspase 9. So I want the product caspase 9, but caspase 9 is in inactive form. How the caspase 9 is coming to active form? You ask the question yourself. Caspase 9 is coming to active form by cytochrome C cytoplasm. Only cytochrome C cytoplasm will combine with APAF, that is apoptotic protein activating factor. But how cytochrome C is coming into the cytoplasm? Only when this channel opens. When this channel opens, stress sensors, which we discussed in previous slide. Okay, now caspase 9 is activated. What caspase 9 does? Caspase 9 will be converted to caspase 3, or caspase 9 will stimulate caspase. That way caspase 9 is initiator caspase, caspase 3 is execution caspase. Extrinsic pathway for apoptosis is also, this is same, caspase 3 is going to be activated. The final product of extrinsic is also caspase 3, which is executor caspase. What caspase 3? It will produce protease nucleus which is going to digest the proteins, nucleic acids and destroy the cell. That is apoptosis is going to take place. In extrinsic, the stimulus is from outside the cell. For example, any infection, let us be as death receptor, this is the domain. This is death receptor and this is domain. So this we call it as tumor necrosis factor, death domain, which will be binding with tumor necrosis factor receptor, extrinsic. Because of some infection, what happens? This tumor necrosis factor combines with tumor necrosis factor receptor, or this will also be better, FAS. This is FAS, FAS, FAS receptor, this is FAS ligand. So just to concentrate, when FAS, FAS ligand binds with FAS receptor, this apoptosis inside the cell is going to be triggered. But for this FAS ligand to bind with FAS receptor, some infection has to occur from outside the cell, like any viral infection. Because of this, what happens? Inside there a domain is created, FADD, FAS activated death domain. F for FAS, FAS, FAS activated death domain. And because of this FAS, so remember, FAS will be coming in extrinsic pathway of apoptosis. The cytochrome C, APAF will come in intrinsic domain, intrinsic pathway of apoptosis. MCQs, it will be asked, maybe note it down. Now what happens? Because this is activated, caspase 8 is activated. So pro caspase 8 is converted to caspase 8 activated. So in extrinsic pathway, for caspase 3 to be activated, I need caspase 8. But for caspase 8 to be involved, I need this FAD, or this FAS has to bind with FAS receptor. FAS to bind with FAS receptor, some infection is needed to be coming from outside the cell. So this is the extrinsic pathway. And again the final output for both intrinsic pathway as well as extrinsic pathway of apoptosis is caspase 3 activation. And caspase 3 we all know, protease nucleus is provided. I will explain that also, how this proteases and nucleases finally produces the destruction of the cell, which is called as apoptosis, programmed cell death. What are the stages? Step by step. So I will draw the diagram so that I can write the stages here. So this is normal cell. Then what happens? The size is reduced, shrinkage, chromatin condensation, bleb, membrane bleb is formed. Then from this bleb I get, so this comes separate, I get apoptotic body, and finally this apoptotic body phagocytosis. So phagocytosis of the apoptotic body or that shrunken cell, the entire cell is being phagocytosed and removed. The process of apoptosis, the cell membrane is intact, whereas in necrosis the membrane is ruptured, and in necrosis there is spilling of contents from the destructed cell and that can infect other cells. Spread is possible, but here the entire cell is going to be phagocytosed, the apoptotic body, even apoptotic body has a membrane intact. That is what I want you to mention, membrane is intact in the entire stages of apoptosis, and the final step is phagocyte. So stages you can tell yourself. First stage is shrinkage, second stage is chromatin condensation, third stage is bleb, fourth stage is body, apoptotic body formation, fifth stage is very important, phagocytosis. We have to ask me the question, why that particular cell is alone being phagocytosed, not the other normal cell? This is the question I am asking you. This is normal cell, this is apoptotic body. So this is phagocyte. Now just a concept, my question is, this phagocytosis is not going to phagocyte or eat the normal cell. The phagocytosis is going to eat the apoptotic body and the cell involved is which is going to be destroyed by apoptosis. How is the question? Now just to concentrate, in normal cell this is called as phosphatidylserine. Going little bit depth, but okay, worth mentioning. So in normal cell that phosphatidylserine is present inside the cell. So the phagocyte is not going to be attracted to the phosphatidylserine. This is one of the attraction for phagocyte, but here it is inside the cell. So this phagocyte is not attracted towards the cell, but whereas here because of this process of apoptosis and all those caspase activation, the same phosphatidylserine is coming to our order and this is being attracted. When the phosphatidylserine is on the surface of the cell, phagocyte loves to attract or engulf that. Normal cell, the phosphatidylserine protein is present within the cell. Once apoptosis takes place, the phosphatidylserine appears on the surface of the cell. That is one of the reason why this phagocyte loves to take the apoptotic body and not the normal cell. That can be asked in viva, please note it down. Now let us go for markers of apoptosis or investigation to find out. So hope this video is very useful to you. It is a very important video and I have covered almost every aspect of apoptosis. So I hope you will be thorough with apoptosis. Before ending the video, I will put a question to you and I want to check whether you are able to answer, and please put your answers in the comment section. Caspase 8, caspase 9, caspase 10. This comes under initiator caspase or execution caspase. First question. Yeah, my second question I am not going to answer, you have to answer in the comment section. In normal cell, cytochrome C will be present within the membranes of mitochondria or in the cytoplasm. So that is my question. So within the membrane or outside the membrane, inside the cytoplasm, you can answer it in the comment section. So hope this video was very useful to you on apoptosis. I have explained in detail what is apoptosis, why apoptosis occur, types of apoptosis, in the mechanism what is intrinsic, what is extrinsic, and factors pro-apoptotic, anti-apoptotic, and how the balance is maintained between the life of the cell and death of the cell. So hope this video is very useful for you, and if you have not subscribed to our channel, please subscribe to our channel Physiology Lectures by Dr. Sendil Kumar, and please share it to our medical students, dental, paramedical, nursing, all the medical community, and if you have any doubts, please don't hesitate to share it in the comment section. Thank you. We will meet in the next video, and if you have not subscribed to our channel, please subscribe to our channel. Thank you. We can meet in the next video. Hope this video is useful to you.